Alright guys, this is going to be Dante, Evan, Jacob, and Brigett, and we are going to do protein synthesis through the use of the magic school bus. The class thought long and hard about what they knew about this protein synthesis. They thought and thought really hard, and even Wanda Lee, the smartest girl in the class, was confused on what protein synthesis could be. Miss Frizzle sees the class struggling and enthusiastically announces to them, "Hey, why don't we take the magic school bus and find out for ourselves?" The bus flew in triumphantly and the class cheered. They all shouted, "Field trip, field trip, field trip!" Miss Fizzle announces to her class that they will be learning about today. "Alright class, are you ready to learn about protein synthesis?" exclaims Miss Frizzle. "It's a marvelous process that makes proteins with the use of transcription and translation. Transcription is the synthesis of RNA, while translation is the synthesis of actual polypeptide."
The class was very excited as Miss Frizzle explained what initiation of translation is, which begins when the 5' end of the mRNA attaches to the small ribosomal subunit at the start codon. Next, initiator tRNA carrying methanoid amino acid with anticodon UAZ base pairs with the start codon in the P site. Finally, the large ribosomal subunit attaches. The arrival of the large ribosomal subunit completes the initiation complex.
Their next stop was the elongation point, which starts at the RNA polymerase as it unzips the DNA. It untwists and shows about 10 to 20 DNA bases that will pair up with RNA nucleotides, basically matching up with its other base pairs. This is where the mRNA is made out of, and RNA nucleotides grow from the 5' end to the 3' end on the other side. Also, remember this kids, but the RNA polymerase 2 reads the sense DNA.
Termination of transcription occurs when RNA polymerase II reaches the termination sequence of AAU, AAA in the DNA. It continues 10 to 30 bases past the sequence, then jumps off the DNA, while mRNA feels completely free from the sense DNA. mRNA processing must occur before the pre-mRNA leaves the nucleus. A 5' guanine cap is added to the 5' end of the mRNA, while 3' poly A tail is added to the 3' end. The cap and tail are important as they help move the transcript out of the nucleus, but they also protect the mRNA from degradation by hydrolytic enzymes.
Then Ms. Frizzle explained in the class that mRNA splicing is the process of removing introns from pre-mRNA. In pre-mRNA, there are exons, which are coding regions for coding amino acids, and introns, which are non-coding regions. In RNA splicing, mRNA is edited by spliceosomes which contain snRNA or SNRPs, and these SNRPs attach to the ends of introns, then more pull the ends of introns, forming the spliceosome, which removes introns and connects exons.
Miss Frizzle then pulled out a very complicated chart. This chart was called the mRNA coding dictionary. Basically, you can see where the where it starts and then where it ends. For here, we have the AUG for the start, and then the UAG or UAA for the end.
So, as a whole, translation was actually the synthesis of a polypeptide by mRNA in the ribosomes? Asked Arnold. "Well, yes," exclaimed Miss Frizzle, "that's exactly right." Their next stop was to analyze ribosomes, which combine specific tRNA anticodons with mRNA codons. Ralphie commented on how it looked like a hamburger and how there was a small and a large subunit. "Well, yes, I suppose so, Ralphie. Both buns are made of the rRNA made by polymerase I and more proteins. They were both made in the nucleolus inside the nucleus," said Miss Frizzle. The children learned how each ribosome had binding sites from mRNA and 3 binding sites for tRNA molecules. The P site held the tRNA with the polypeptide chain, the A site carried the tRNA with the next amino acid that was going to be added to the chain, and the E site is where it exits or leaves the ribosome.
The tRNA structure basically has 80 nucleotides which fold into a 3-dimensional shape. They also go attached to the 3' end of the nucleotide side.
Aminoacyl tRNA synthetase is a special enzyme in cytosol that connects the proper tRNA to its correct amino acid. There are 20 different enzymes for each of the 20 different amino acids.
Mrs. Frizzle then went on to explain what initiation of translation is. Initiation of translation starts when the 5' end of the mRNA attaches to the small ribosomal subunit at the start codon. Next, an initiator to get tRNA is carrying a methanoid amino acid with anticodon UAC. Base pairs with the start codon and the P site. And finally, the large ribosomal subunit attaches and completes the initiation complex. Proteins called initiation factors are required to bring all of the translation components together.
Elongation of translation, Ms. Frizzle said, was like a conveyor belt with 3-step cycles, as each amino acid is added to the next one. The A site is empty, so a tRNA molecule with the second amino acid can land on it and fill it up. The tRNA anticodon has to be paired with its proper mRNA codon and the A site to make sure that the correct amino acid is being added. After that, peptide bonds development occurs as the bond between polypeptide and the P site joins the new amino acid in the A site. The tRNA at the P site from the growing polypeptide chain. This step separates the tRNA at the P site from the growing polypeptide chain and transfers the chain, now with the extra amino acid, to the tRNA at the A site. As a whole, tRNA from the growing chain of polypeptides moves the chain with the added amino acid to the RNA in the A site. tRNA in the A site. During translocation, the ribosome moves the tRNA with the attached polypeptide from the A to the P site. The next codon is now available at the A site, and the tRNA in the P site is then moved to the E site, or the exit site, to leave the ribosome. Translocation makes sure that the mRNA is read from the 5' to 3'. And the 3 steps of elongation continue to add amino acids until it's finished, all within 1/10 of a second.
Termination of translation occurs when a ribosome reaches a stop codon on the mRNA. The release factors hydrolyze the bond between the tRNA and the P site and the last amino acid of the polypeptide chain. The polypeptide is then released from the ribosome. The 2 ribosomal subunits and components of the assembly dissociate. Everything is then reused.
Amino acids consist of amino group, a central carbon, a carboxyl group, and an R group. They're all the same except for the R group is responsible for determining which one of the 20 amino acids it is. The peptide bond formation has rRNA in the large ribosomal subunit, which is like a ribozyme, to join amino acids to make a peptide bond through dehydration synthesis. The peptide bond forms between the carboxyl end of one of the amino acids and the amino group of the other one. Water is removed as the bond is formed.
Polyribosome is a single or as many ribosomes translating into a single mRNA molecule. Basically, this just allows for proteins to be made very quickly.
Protein structures have 4 levels. Primary structure is the sequence of amino acids in the polypeptide chain. Secondary structure is loops, coils, and sheets that are formed by hydrogen bonds between non-adjacent carboxyl and amino groups. The tertiary structure is the 3-dimensional form held together by interactions between the R groups and the water by electrical charges. Finally, Ms. Frizzle explained that After translation, the polypeptide chains undergo modifications which take place in the Golgi apparatus. Sugars, lipids, and phosphates are added onto some amino acids, while other amino acids may also be removed. Protein's three-dimensional shape can also be modified.